So our next speaker is my co-host, Dr. Margot Cousin, a familiar face in the room today. So welcome back.
I'm really excited to talk to you guys today about really our process for developing an ASO for ADLD. And I'm not going to get into really nitty-gritty data as that's still being generated with our collaborators, but really talk about the process and where we are. All right, so I'm not going to review really the mechanisms of ADLD. I think that's been covered as I anticipated it probably would be. But we'll talk a little bit about antisense oligonucleotide therapies or ASOs, how they work, and why we think they're a good choice for ADLD. I'm going to talk about our partnership with the n-Lorem Foundation. And then a little bit about our specific progress in the development of that ASO. And then in the last little bit, talk briefly about what we're thinking for clinical trial readiness and how do we get ready to move these trials forward. Not going to go into this data that it was beautiful from Dr. Patiat about. We understand that this is largely a gene duplication that leads to overexpression as the mechanism, and that we've seen beautiful data that shows that by lowering the levels of expression, we can recover more normal phenotypes in cells, and beautiful data from Dr. Patiat showing that the animals are doing better too. So what are ASOs? So ASOs are a gene-targeted therapy. They're essentially short segments of single-stranded nucleotides like DNA or RNA, often between 13 and 30 nucleotides long. And they target the RNA product of the gene. And the goal is to correct or overcome the genetic defects by this ASO interacting with that RNA through sequence-based Watson-Crick base pairing. So we do not alter the DNA, so it's not a gene therapy or a CRISPR therapy where we're editing the genome at all. So it's purely a transient effect. These patients, if this is effective, will have to get repeat dosing to maintain that therapeutic effect. They are chemically synthesized. This is not a biologic. It's just a moderately large molecule. But they're modified in several different ways to increase the drug-like qualities of the oligonucleotide. So this means we can increase the stability, we can evade the immune system reactions, we can increase cellular uptake and optimize kind of target affinity. So a little more granularity. I'm not going to go into great detail here, but in the middle we have the two nucleotides. So it goes from 5' at the top to 3' at the bottom. You can see the ring that is the sugar. The base is off to the right. We have the ACGT highlighted at the upper nucleotide. And at that 2' position where it says R at the top, we know that this is what defines DNA or RNA, whether it's a hydrogen or a hydroxyl group. And substitutions at that position can change how this oligonucleotide will function. We can replace this with a methoxyethyl, which is a common substitution, and this can increase our affinity to our targets. Again, decrease the nuclease's ability to degrade these molecules, and it can also affect downstream function. We can also conjugate these to molecules like a GalNAc, and many others are in development to target specific tissues. This wouldn't be necessary for a central nervous system disease as we administer it directly into the CSF through intrathecal delivery. But this can be done for other targets. And then lastly, the linkages between the bases can be modified. So we normally have the phosphodiester linkages, and we change these often to a phosphorothioate that changes the charge a little bit. Also prevents immune system interaction, decreases, again, the nuclease degradation, and helps with cellular uptake. So a couple of different mechanisms by which ASOs can work. And in this figure, we're lumping siRNAs into the ASO category, which is also true, but we're not going to talk about those today. But essentially, there's gapmers and siRNAs that both target that RNA that you're targeting for degradation. So we're going to basically decrease the amount of gene expression by decreasing the RNA of that gene product that we're targeting. The gapmers, which is what we'll talk about today, they're called that because the middle part, if you look on the left on the top, is DNA. So it's deoxynucleotides in the central portion that allows for when we interact with the RNA target, we get a DNA RNA heteroduplex. And that is recognized by this enzyme called RNase H1, and it will degrade that transcript. And then also called a gapmer, because the outside nucleotides have those full modifications that I talked about that maintain that drug-like quality for that molecule. So we can still have higher levels of stability, we can get good cellular uptake and those other qualities. So these are the molecules we're going to talk about today. A little more simplistic design here again. So we have our DNA, that's going to be our gene of interest. This is transcribed into RNA, and that's where our ASOs are going to target. And then depending on your mechanism of disease, we want to alter how that protein is made or how it functions. So if we think about ADLD, we know we have generally an increase in the number of copies of Lamin B1 or a deletion that leads to overexpression. But either way, we get Lamin B1 overexpression at the RNA level that leads to that Lamin B1 protein accumulation. And our goal is to treat these patients with an ASO that can interact at that RNA level to decrease RNA expression that will lead to a decrease in protein production. And this is the mechanism that we hope to be therapeutic in our patients. So from here, we know that ASOs have been approved for many different uses, right? So we understand the chemistry. We know generally the safety profile, the side effects, and how to administer these drugs. And what's unique about ASOs is that they're considered a platform therapy, in that we can change the sequence of just the bases of those nucleotides, right, to change it to any target of interest, but we're not changing the chemistry. So we don't have to go back and maybe do the full rigorous safety tox studies when we're thinking about the FDA, especially for these severe diseases, because we understand how these are working. Small molecules don't behave that way. And most of other gene targeted therapies, we're changing too many things, right? We have a gene vector, we have adjuvants and delivery mechanisms that are going to play a role in how this drug is developed. So there's some unique attributes with ASOs that allow us to translate these potentially more rapidly. And there are many in clinical trials right now as well to say that they're quite versatile and able to target many different genes. I always have to talk about TimU's study. This really changed for sure how I think about what we can do for therapies for rare disease patients, but I think also in the field really has been a paradigm shift. So TimU, for those who don't know, is a investigator out of Boston Children's Hospital, and he saw this little girl, Mila, who had Batten disease. And they had to do genome sequencing in order to get her genetic diagnosis. And after realizing what the cause was able to design, develop, test in her cells, and work with the FDA to get a sort of a hybrid compassionate use research IND to treat her with an experimental ASO in under a year. No one has brought a new drug to market in such a rapid time frame for a rare disease patient like this. So what we can do is different now than maybe what it was even just a handful of years ago. So from that sparked a lot of work and a lot of interest in thinking about ASOs for rare disease. And the FDA has issued some draft guidance since that time to help us determine how best to do this, right? And thinking specifically about these individuals that have these severely debilitating and life-threatening diseases. You know, for Mila that TimU treated, and for many others, you know, we understand the risk of not treating these patients. It's dire, right? And the risk of not treating is often much greater than the risk of treating with an experimental drug like an ASO, where we have, you know, pretty well-proven safety profiles of the chemistry and we know how they behave. So that gives us the opportunity then to get there quicker and reduce maybe some of the safety and toxicity studies to balance the time and workload going into making one of these drugs and getting them to the patient in a time that we can actually maybe help them. So kind of summary of this part, right? We know that ASOs, like, why are these good for ultra rare and N of 1 diseases? They can be rapidly developed. Again, the chemistry is well understood. It's considered a platform therapy. And I say cost effective, but more cost effective than a lot of other drug development opportunities, right? In addition, these are extremely stable. So, you know, we can make one batch of an ASO we can make at one time is probably enough to last that patient their entire lives, and it can sit on the shelf for as long as they need it. So if we think about other potential therapeutic modalities that might have to be remanufactured, That just isn't the case here. We also don't have adjuvants, delivery vehicles, or some of these other considerations to worry about that can really significantly increase the burden of your manufacturing and development processes. And then lastly, we have FDA guidances and regulatory support to help us move forward. I also wanted to mention we're working as a part of the N=1 collaborative. This is largely an academic group, but bringing together folks from across the spectrum of rare disease and industry and advocacy to define best practices. So working with the FDA, working with other groups to say, how do we do this as safely, as quickly, and as equitably as possible to get ASO or other types of individualized therapies to the patients in need. And they've been instrumental in helping a lot of different academic programs and others and patients find the right resources to move these programs along. And I've received some slides to share with you from our partners, the n-Lorem Foundation, that I think can highlight some of the benefits of this opportunity. And then I'll walk through kind of where we are in this process. So the n-Lorem Foundation was started by Stanley Crook, who was the former CEO of Ionis, who was the pharmaceutical company that has put out most of the ASOs on the market today, or many of them. And after Tim Yu's work, and others were really gaining a lot of motivation, I think he realized that there's a lot of opportunity here and they could apply the vast knowledge that they've gained from being a pharmaceutical company developing ASOs and figure out how to tweak that process to make smaller amounts to focus it on an individual patient, but apply that same rigor that they've been able to learn about through his work in the pharma industry. So their mission is to apply the efficiency, versatility, and specificity of ASOs to treat patients with what they consider nano-rare diseases or less than 30 patients worldwide. And we certainly understand the desperate situation of patients with these ultra-rare, ultra-severe disorders where there are no options. It often takes a long time to even achieve that diagnosis, and then once they get it, they realize there's no specific or curative therapy available. And then looking to the resources that are available to make drugs and to help patients in this situation, there's really not a good commercial avenue to pursue for these ultra-rare conditions. I think we're getting better with, rare designations and some of these other mechanisms that they're coming up with to help promote development for these ultra-rare patients, but it's not yet scalable for these conditions. So is a nonprofit model what's required? Somewhere between nonprofit and academia seems to be the way that these are going forward right now. And again, the reasons I discussed previously is why n-Lorem has feasibility in their approach, right? All of these attributes listed here make this potentially scalable, or at least more scalable than a lot of other drug modalities, right? We're looking at the same type of chemistry, the same type of drug manufacturing. We're just changing it to look at different targets. So if you can ramp up your screening and your in vitro data for figuring out if your targets are working and optimizing your selection, then the downstream parts should be fairly similar. And they've been really good about publishing a lot of what they're doing. Hopefully we can, with n-Lorem as well as with the N=1 collaborative, really pull these patients together so we can continue to learn about this process so we can do better for all programs. What side effects, what process, how do we design clinical trials? How do we move that needle forward so we can, every new patient that we treat, do a little better, right? Learn a little more and have more information, optimizing their drug and their trial so we can see benefit. And again, their work of industrialization of the experimental ASO treatment to maximize the quality and maximize the learnings, which is what I've kind of already said. And I think importantly, it always takes a village. Oops, skip the slides. So they've amassed quite a number of collaborations with academic centers like Mayo and others, as well as with industry, manufacturers, and across the board to make this process as cost effective and as efficient as possible. And this is the same groups of companies and endeavors that many of the academic institutions are also partnering with for their own ASO development, as are we. So here's a list of the applications and submissions that n-Lorem has received. You can see all the different genes here representing those disorders in patients, and the numbers that they have accepted so far. And you can see that Lamin B1 is on the list. And for n-Lorem's data to date, for those patients that have been treated and they have data available, so 2 patients were lost to follow-up, but 5 of 5 evaluable patients, they say that they've shown evidence of clinically important benefit, and for 6, we're too early to tell yet. So pretty positive findings for a very experimental paradigm. And importantly, publishing a lot. So again, pulling that data together, so hopefully we can do better in the future with what we're learning for these programs. All right, so what I really want to talk about are the progress we're making for the Lamin B1 ASO. So this is the outline of really n-Lorem's process for screening and designing and testing ASOs. And if I look on the left, we have what's considered the screening steps. So each one of these are a different step in the process of getting to that lead ASO and testing it. And then the far right columns, second to last, is what they typically do for their programs. And these are all around, again, those gapmer ASOs to degrade the transcript of interest. And on the right, the status of the Lamin B1 ASO. So really first step is to design many different potential ASO targets and assess for any off-target effects. And this has been completed quite a bit ago. And then that primary ASO screen looking for knockdown. And what their minimum criteria is 80% target reduction. They typically screen around 500 ASOs, and for Lamin B1, they looked at 624. From there, they move on to dose response evaluation, looking for an IC50 of at least 1 micromolar that's given via just free uptake in the cells. Typically between 50 and 75 ASOs is what they screen, and they looked at 55 for Lamin B1. Then moving into in vitro off-target analysis. So based on the sequence, we can predict what likely off-targets might be based on where this ASO could interact with given, mismatches or similarity in sequences to the target. And they go back and actually look at those and see which have changes in the expression levels in the cells that they're testing. And that was completed and ruled out that there were any issues with those. Then we move on to what we're calling the BJAB assay. That is referring to the cell line that's used to measure. But this is looking at the innate immune response. So there are some sequence characteristics and also some non-sequence based characteristics that may lead to activation of the innate immune response. We want to rule those out. So they typically screen between 50 and 75 ASOs. And that was completed and moving forward with only those that did not activate the immune system. Then we move into single dose tolerability screening in rodents at high doses and includes histopathology of the central nervous system, different cell types. We typically look at about 20 ASOs, and here we're able to identify 4 that were really well tolerated in mice and 3 also in rats. And from here, they take all this data into account and try to figure out what your lead compound is, which one maximizes benefit and has the most reduced amount of potential off targets or safety issues predicted, and move into the IND enabling toxicity studies in rodents. And this for our Lamin B1 ASO is near complete. We're maybe days away. We anticipated that being about the middle of July, so we're almost there. And importantly, while that toxicity study is going on, that lasts about 3 months, they're doing all of the GMP manufacturing and the sterile fill and finish to produce that drug product ready to go. So as soon as we're ready with those toxicity studies, we're betting on the fact that they're going to come back clean. We can immediately submit that IND application. So if we look at the process kind of in total, a more graphical view, we can walk through what these steps are. Often for a knockdown ASO, like a gapmer, we're going to want to do genome sequencing of the patient DNA because we might do allele specific. We're targeting a specific mutation. That is not the strategy we've taken here. So this isn't knocking down just one allele of Lamin B1. We're just knocking it down in general. So we didn't have to do that part of the work here. Moved right into the ASO discovery phase, then selected the lead. and then moved in again into the 13-week GLP toxicology study that's in parallel with that manufacturing and sterile fill and finish steps. And again, we're almost at the end there, in which case we'll finish putting together our IND application and submit. Following FDA approval, we would then seek approval through our IRB and begin our patient treatment in an N of 1 trial. And I need to spend just a moment to talk about this blue arrow down here, which is understanding what's going on with our patient, establishing that clinical baseline, and determining what it is we're going to measure in this protocol to try to show benefit. The primary outcome we're looking for in this study is safety, right? That is number one. We want to show that this is safe. We want to understand if there's any side effects, if there's any concerns with this drug. We do hope that this can be made available to additional ADLD patients. And this is really step one to understand what that might look like with the specific ASO. But this is an opportunity for us to also try to measure benefit, right? As we know, this is a slowly progressive disease, and that's a challenge. You know, how do we in 1 to 2 years hopefully show a change in trajectory for this patient if there is one? You know, if there is one, we need to capture that information. So we've been partnering with the Center for MS and Autoimmune Neurology here at Mayo. We're working closely with Department of Clinical Genomics, Dr. Demija, and others to really rigorously phenotype our patient and do these clinical assessments to capture as much as we can. Still trying our best to stay close to standard of care. So this is applicable and generalizable to others, but also trying to capture as much granularity as we can. So if there is a change, we can see it and measure it. So there's been a lot of effort in figuring out how that protocol should be developed. And I hope the next time we get to talk about this, I can share more details of what we're doing and how we're doing it, and hopefully some results. So that takes me into thinking about clinical trial readiness. So a lot of that is that patient disease baseline and establishing those endpoints. You know, looking at MRI and gait and movement assessments, autonomic function and others. And again, I have the groups here at Mayo that are really helping us do that well in developing that protocol. But what else is there, right? We want to translate this to more than just one patient. So we're also thinking about biomarker development. We're working with our proteomics core and have a project underway with the Department of Neurology to think about CSF and try to capture, as we will be able to get CSF over time for this patient, try to see what's changing, right? What's associated with disease and what's associated with treatment. That's going to take also getting other patients and getting other samples to continue to move that effort forward to really gain the data we need to determine those biomarkers. And then in order to translate this, we really need to understand these patients as a population. So, and move from not just the clinical descriptions, but endpoints and clinical outcomes assessments that we can use in clinical trials. So I'm really excited to announce that we're partnering with the Children's Hospital of Philadelphia and the Global Leukodystrophy Initiative to start a natural history study. This should be opening up for enrollment very soon, and Dr. Demija is going to talk a little bit more about this later. But that this will start with chart review type study, and hopefully we can show some good evidence moving forward that we might be getting funding for a more prospective study that will allow us to again establish those clinical outcomes assessments that are going to be so key. Also will hopefully allow us to validate biomarkers and really engage with the population of individuals affected by ADLD so we can understand who wants to participate in a study like an ASO trial and how we could move that forward. So with that, there's lots of people to thank. For sure, all the team in SIM for helping support moving the ASO work forward in general and specifically for ADLD. Our clinical partners who've been working with our patients and helping us understand how we marry the translational pipeline to clinical practice. The CMSAN team who's been instrumental in helping us describe our patients and measure these really intricate and important outcomes and phenotypes. Many collaborators who informed along the way. And then for sure the support and n-Lorem Foundation for their partnership and support, ADLD Center, the Center for Individualized Medicine, CCATS here at Mayo, and the Center for MS and Autoimmune Neurology. So with that, I'd be happy to take any questions.
Initially, but does the immune system recognize them? Are they recognized by MHCs or? Generally,
there's not much of an immune system response. There is some innate response to ASOs, and often that has to do with sequence based. So triple G's and C's can help elicit that response associated with a few different receptors. It isn't a large problem, but it's certainly one that we want to minimize, right? And we usually through that BJAB assay they've shown that it correlates well with human studies that have had an immune response associated with them. So we think we can predict it. We think we can minimize that. So that's not an issue using those assays.
So generally, do you know how frequent you would need to give them ASO, like repeated before you start treatment or as you go? Is that question?
For developing a new?
Yeah,
Yeah,
like how,
so
they would have to be given, you said like repeated, right?
So generally the paradigm, you're going to have more close together dosing as you start with dose escalation. So those are typically a couple of weeks or a month apart. And then once you get to a maintenance dose, it's usually quarterly, right? So we're looking at every 3 months for a central nervous system delivery. If it's a different tissue, it's going to have different route of administration and different timing of that administration. So we can deliver to the lungs, the liver, the kidneys, and the eyes, and they all have a different frequency, different dose that we do deliver. But yeah, quarterly is what we think for.
So it's not so much disease dependent, it's tissue kind of specific, the frequency was my question.
That's generally how they do it. I know for some programs, it's changed, right? So for some of the patients that have had success so far, right, there are a lot of seizure disorders, and they can see really easy with an outcome like a seizure when the drug starts to wear off, right, when the seizures start to break through. So I think they've either adjusted that slightly by changing the dosing schedule or by increasing the amount that they're giving in each dose. But to my knowledge, they try pretty hard to stick to that quarterly regimen. But I'm not aware of all the specific programs of what's going on.
Okay. So since this gamma ASO isn't targeting a specific variant within the Lamin B1 gene, it's targeting a duplication instead. How are you regulating how much downregulation you have of this gene back to wild type levels versus just fully knocking it out? Is it dosage or what is it?
Great question. So the original screen is looking at greater than 80% knockdown in cells, right? But that's obviously going to be dependent on how much you can deliver to that cell. So the hope, well, a couple of things, right? As Dr. Padilla had talked about, we think that the levels of Lamin B1 generally drop pretty low in mature cells. So, you know, we don't believe that there's a great risk of the too low effect necessarily. But we would expect to manage that if we think that that is a problem through dosing and timing of those doses, as opposed to tweaking the design of the ASO, for instance. Yeah. I think there's Radhika, do you have a
So how are you going to measure kind of target engagement? Like, are you going to look at CSF Lamin B1 levels? How do you know if the ASO is actually working or not?
Great question. And that was one of our questions early on as we were designing the trial and trying to figure out what we need to do. There are methods to develop the ASO in CSF to know that we're delivering it. And we haven't yet shown if we can measure RNA levels in CSF. That's something we would like to do, but we haven't done that yet. So right now there is no plan to actually look at specific target engagement in our patient. We are hoping to look at, you know, neurofilament light and GFAP and other markers of neurodegeneration, but there's no direct measure to show that right now. I think as we would move toward a larger trial, that's probably going to be a much larger consideration. But right now we're looking at safety as being number one. Yeah. Online questions?
There is one more question actually. How are we selecting the first in human dose for the SLO? Will this be scaled down from the max tolerated dose?
Yep, it'll be informed by the toxicity studies, but also by the many other programs that have been given ASO doses in the past. So I think that's a conversation we'll have to have once we get that tox data back from the GLP study.